Hello everyone, today we are here to discuss about the previous year NEET questions of digestion and absorption. This is the part 3 video. So going on to the questions. Select the correct match of the digested products in humans given in column 1 with their absorption site and mechanism in column 2. So the first option is fructose and Na+, the small intestine by passive absorption. So we know sodium is the carrier ion, and this happens in the small intestine, but sodium is transported actively. Glycerol and fatty acids, duodenum moved as chylomicrons. No, duodenum is where the digestion happens, so most of the absorption occurs in the jejunum and ileum. Okay, so it occurs mostly in jejunum and ileum. So here also it's the same, it occurs in the posterior part, the other parts of the small intestine. Cholesterol and maltose, large intestine active absorption. No, mostly in large intestine, water, alcohol, some drugs, such things are taken up, and it mostly large intestine functions to stiffen the stool, that is to harden the stool, like it absorbs the water out of the stool and makes it hard. Okay, so glycine and glucose, small intestine active absorption. Yes, correct. So glucose is by active transportation, glycine and glucose both by active absorption, that is by transport of ATP. Okay, so the option is option D. So the solution is small intestine is major area of absorption of nutrients. Approximately 80% of absorption takes place here. Glucose, fructose, fatty acids, amino acids, glycine, etc. are absorbed through mucosa into the blood and lymph by active absorption. Okay. So glycine is an amino acid. Noted. Point to be noted. A healthy person eats following diet: 5 gram raw sugar, 4 gram albumin, 10 gram pure buffalo ghee adulterated with 2 gram vegetable ghee. So vegetable ghee is vanaspati. Okay, vanaspati, we need to know vanaspati, unsaturated fat. So, and 5 gram lignin. How many calories is he likely to get? So we need to know the physiological values of calorie. Okay, so we'll see what are the values. Oh, we have it in the solution, we'll see. Okay, of carbohydrate, so we know raw sugar, it's like glucose, so we can translate it as carbohydrates. Okay, so it's given as 5 gram, so we have 4 kilocalories per gram, so we have 5 grams here. Albumin, what is albumin? Albumin is the white portion of the egg. So in egg we have the yellow portion and the white portion, and white portion is the albumin and yellow portion is the yolk. Albumin is the completely a protein. Albumin is also present in our body in extracellular fluid. So here, since we are talking about diet, it refers to egg albumin, which is protein. Okay, and we have buffalo ghee adulterated with 2 grams of vegetable ghee, which is vanaspati. Okay, so fat, it's given 9 kilocalories per gram. And lignin, what is lignin? It's a plant substance, that is, it is present in the walls of the cells. So the calculation part is here. We have 5 grams, so it's 20, 5 into 4, 4 grams of albumin, which will yield 16 calories. 10 plus 2, yeah, I told you, like this is unsaturated, this is also unsaturated. If it's given vegetable oil, we can consider other things, but it's given the hydrogenated vegetable oil, we know vanaspati, so it's same as animal fat only, so that is also taken into consideration. What happens to the 5 gram of lignin? Lignin, it is not metabolized to get calories, because we don't have any digestive enzymes to break down lignin. Okay, so the grand total is 144. So the answer is 144. Which enzymes are likely to act on baked potatoes eaten by humans starting from mouth as it moves down the alimentary canal? Okay, we know mouth is where the digestion starts. So we have been given the options: salivary maltase, carboxypeptidase, trypsinogen; pancreatic amylase, salivary amylase, lipases; disaccharidases like maltase, lipase and nuclease; salivary amylase, pancreatic amylase, disaccharidase. So potato is basically made up of starch, so we have to consider starch. What is starch? Broken by amylase. So we eat in the mouth, so 30% of the food that we eat, that is starch, is digested in the mouth. Fact to be remembered by ptyalin, which is the salivary amylase. So digestion of potato starts in the mouth, so it basically has to be salivary. So we have two salivary options, and one is maltose and one is amylose. So here we consider amylose because it's starch. So the answer is this. And then pancreatic amylase acts, and then disaccharidases act. So through rejection we can easily follow amylase, disaccharidases. Since this is starch, we have to follow amylase only. Okay. Chemical process of digestion starts in oral cavity by hydrolytic action of carbohydrate splitting enzyme, the salivary amylase. Carbohydrate in the chyme are hydrolyzed by pancreatic amylase into disaccharidases. Okay. So the answer is option D, salivary amylase, then pancreatic amylase, then disaccharidases. Which one of the following enzymes carries out initial step in digestion of milk in humans? This is a repeated question, so we know it's the renin. Trypsin acts on protein, lipase acts on fats, trypsin acts on proteins again in the duodenum. Renin is an enzyme that is essential for protein digestion. It's active in acidic medium. That's why we have the HCl. Okay. So the answer is renin. It's present in children. One of the constituents of pancreatic juice while poured into the duodenum is. It's a tricky question, so like we generally tend to make a mistake. So we have chymotrypsin, trypsin. So we might think both options are right. No. So we know that when enzymes are being poured into the duodenum, they are poured as inactive gens. So trypsinogen and chymotrypsinogen. So enterokinase is produced by the duodenum epithelium mucosal cell itself, that activates all the trypsinogen and chymotrypsinogen. So the answer is trypsinogen. Trypsinogen is an active. Trypsinogen is an inactive form, and we know that all enzymes of pancreas are secreted in this form. An enzyme enterokinase is secreted by the duodenal mucosa itself at chemical stage that changes the inactive pancreatic secretion trypsinogen to chymotrypsinogen. So the trypsinogen becomes trypsin. The trypsin in turn activates chymotrypsinogen to chymotrypsin, and hence the process continues. So the answer is trypsinogen. A young infant may be feeding entirely on mother's milk, which is white in color, but the stools which the infant passes out is quite yellowish. What is the yellow color due to? Okay, this is a application oriented question. We know, like we generally tend to see the milk and we'll go only to renin, but we have the infant and renin, and we have the stool color here. We know that bile pigments confer color. So what are bile pigments? Bilirubin. Okay, these confer color. So undigested milk, casein. No, that's not a right one. So like if it's undigested, mostly the child tends to vomit it out. Pancreatic juice poured into the duodenum. No, digestive enzymes, they generally finish their function and they do not give any color. Intestinal juice also, they do not confer any color. So it's generally bile pigments. So like another option which can be given is, maybe they'll replace an option and give bile salts. So what do bile salts do? We get confused if we are given bile salts. Bile salts, we know sodium taurocolate and glycocolate. Okay, so these both give, they do not confer any color, they just break down, emulsification. Okay, emulsification of fat, larger fat droplets to smaller fat droplets. Okay, so bile pigments give the color. So even though the child is taking only milk, the bile secretion is going to be there. So the answer is option A. Sorry for overwriting. The young infant may be feeding entirely on mother's milk, which is white in color, but stools which infant passes out are quite yellowish because the bile pigments pass through bile juice. Bile pigments are any of several colored compounds derived from porphyrin. What is porphyrin? We know RBC, right? RBC has this porphyrin ring, this ring structure. So we know the lifespan of RBC is 120 days. So once 120 days is done, it goes to the spleen, and there it is the porphyrin ring is metabolized, and then we derive biliverdin and bilirubin. Bile pigment is produced regularly when old blood vessels is broken down, mainly by the spleen. So spleen is called the setting bed or graveyard of blood cells. Blood disorders where red cells are destroyed, more pigment is produced. So any question with regards to the color of the stool, we have to consider bile pigments. Which one of the following statement is true regarding digestion and absorption of food in humans? So the options given are fructose and amino acids are absorbed through intestinal mucosa with the help of carrier ions like sodium. Chylomicrons are small lipoprotein particles that are transmitted from intestine into blood capillaries. About 60% is of starch is hydrolyzed by salivary amylase in our mouth. Oxyntic cells in our stomach secrete proenzyme pepsinogen. Okay, so we'll start with the wrong ones. Fructose and amino acids are absorbed through intestinal mucosa with the help of carrier ions like Na+. So this is correct only. Chylomicrons are small lipoproteins that are transported from intestine into blood capillaries. So this is more or less right, but we have only one option. So we have to go for the most apt option. So chylomicrons are generally small particles, right? We have the lipoproteins, that is they have the fat particles at the center, and we have the proteins to pass through the intestinal mucosa, but they generally get into the lymph only. And here about 30% of the starch by salivary amylase. What is salivary amylase? Also called ptyalin, PTAYLI, ptyalin. Oxyntic cells in our stomach secrete proenzyme pepsinogen. No, oxyntic cells produce HCl. Okay, so the answer is option A. Answer is option A. Fructose and amino acids are absorbed through intestinal mucosa with the help of carrier ions like sodium. Which one of the following pairs of food components in human reaches the stomach undigested? So we have to consider what enzymes are present in the mouth. Okay, so we know that in saliva we have the amylase, basically amylase only. So starch gets digested, not totally digested. So we know 30% get digested, 30% is digested. So it does not acquire the same form as it enters the mouth when it enters the stomach. So starch gets modified. So we have to see starch, in which all options we have starch, we can eliminate them. And fat, we don't have any lipases in the saliva, so fat also reaches the stomach as such. Starch is delivered to the stomach in a modified form, but fat, it reaches the same. But we know the three options has starch, so we can't select any of them. But cellulose is a plant, it's present in plant substance. So cellulase is not present in our in saliva. Fat, we don't have lipases also, so the option is fat and cellulose. Fat and cellulose is the pair of food components in human that reaches the stomach totally undigested. What will happen if secretion of parietal cells of the gastric cells is blocked with an inhibitor? So we know the stomach has two types of cells. One secretes the HCl and the other secretes the enzymes required for it. So the options given are gastric juice will be deficient in chymosin. Gastric juice will be deficient in pepsinogen. In absence of HCl secretion, inactive pepsinogen is not converted into active pepsin. Enterokinase will not be released from duodenal mucosa, and so trypsinogen is not converted into trypsin. So what do the parietal cells secrete? We know the parietal cells secrete HCl. Okay, so the chief cells produce pepsin. So in absence of HCl secretion, inactive pepsinogen is not converted into active pepsin. So this is the option. Enterokinase is all secreted by the duodenal mucosa, so it will take care of its own secretion, so that's not a trouble. Okay. If secretion of parietal cells in gastric parietal cells of gastric glands is blocked with an inhibitor, in absence of HCl secretion, inactive pepsinogen is not converted into active pepsin. Okay. A person who is a person who is one along hunger strike and is surviving only on water will have options given are less amino acids in his urine, more glucose in his blood, less urea in his urine, more sodium in his urine. So the person, like, they haven't given any pathology, like the person is not they have not given any particular disease. So in urine we generally have only water and urea, and we also have creatine. So like, let's basically remember H2O and urine. Glucose is not excreted in a normal person. Amino acids, proteins are also not excreted in normal persons. So we can eliminate the option of this, less amino acids in his urine. So in a normal person, proteins do not go out in the urine. More glucose in his blood. So he is on hunger strike. So the first line of substrate that will get depleted is blood glucose. So it should be less. So this option can be eliminated. Less urea in his urine. Why less urea in his urine? What is urea? Urea is the substrate that is formed due to the reactions of many other carbohydrates and other stuff. So since we don't if the person is on hunger strike, we don't generally give any food to him, so that there is no food to be metabolized, so there will be no urea. So this option is correct. More sodium in his urine. So this is also not possible. Sodium also will not be excreted in his urine. So the answer is less urea in his urine. Due to a long hunger strike and survival on water, a person will have less urea in his urine because urine is delivered to kidney as a waste product from liver, which is formed from breakdown of protein, fats, carbohydrates during the hunger. Okay. Lysozyme that is present in perspiration. What is perspiration? It is sweat. Okay, sweat. Perspiration is also called sweat. Saliva and tears destroys. Lysozyme is antibacterial. So certain types of bacteria, all viruses, most virus infected cells, certain fungi. So since it's antibacterial, so the answer is certain types of bacteria. Lysozyme is an antibacterial agent which is secreted by major salivary glands. So it's antibacterial agent. Okay. Epithelial cells of intestine involved in food absorption have on their surface A. Pinocytic vessels, B. Microvilli, C. Zymogen granules, C. Phagocytic vessels. So since epithelium in small intestine has this epithelium, we have microvilli, villi on top of which are microvilli, brush bordered epithelium we say, So this is the microvilli. This will enhance the surface area. So the answer is microvilli. Small intestine is majorly for absorption, so it has to increase the surface area of absorption, and so microvilli. Presence of microvilli on the epithelial cells of intestine increases the surface area for absorption of food. Secretin and cholecystokinin are digestive hormones that are secreted by the duodenum. So secretin and cholecystokinin are secreted in the duodenum. We have to remember the fact. So what is the stimulus that produces this enzyme? Secretin, acidic chyme that enters into the stomach. We have the acidic chyme that enters into the duodenum. So when this stimulus occurs, that's the production of secretin. We have cholecystokinin. It's also called CCK, pancreozymin. Okay, pancreozymin. This is the full name of the enzyme. Presence of fat in the duodenum. Okay, so like as the chyme enters, there's going to be fat in the food. So this CCK, This will, we know that CCK, what it generally does is it does the contraction of the gallbladder, so that when it contracts, the bile just pours out into the duodenum. We know bile acts on the fat, so we know when fat is present, the cells will automatically produce pancreazyme to compensate for more bile. What is their action? Secretin releases bicarbonates into the pancreatic juice, contracts the gallbladder to release bile, stimulating pancreas to secrete and release digestive enzymes into the pancreatic juice. So the answer is duodenum. Duodenum has characteristic Brunner's gland which secrete two hormones called kinase estrogen. The options given are kinase estrogen, secretin, cholecystokinin, prolactin, parathormone, estradiol, progesterone. Estrogen, it's generally a sexual hormone, so it's not found in the intestine. Secretin, cholecystokinin is the answer. Prolactin is generally found in the pituitary gland. Parathormone is generally found in the parathyroid gland. Estradiol is name of a contraceptive. Progesterone is female sexual hormone. Okay, so the answer is secretin, cholecystokinin. Secretin released by duodenal cells inhibit movement in the gastrointestinal tract. Additional fact is it inhibits movement in gastrointestinal tract. As we saw, no, it like when the chyme enters into the duodenum, when more chyme enters, the duodenum will suffocate. So what it does is secretin is produced and it will inhibit the GI movement. Cholecystokinin stimulates release of pancreatic juice. Prolactin is a hormone by anterior pituitary hormone. Parathormone is released by parathyroid glands. Estradiol is released by ovary. Progesterone is released by corpus luteum. Okay. During prolonged fasting, in what sequence are the following organic compounds used up by the body? So this is a fact we have to remember. So we have to search for carbohydrates firstly. So the first line that will get depleted is carbohydrate, and next it's fat, and next lastly it's protein. That's why, like, when you fast, when we do a fasting, first we feel hungry, and then the fats get depleted, and then you can see that there is a reduction in body mass. The person gets lean. That is due to the wasting of muscles, also called as protein is getting spent. That is why we see in marasmus, kwashiorkor, there's a wastage of muscles. Carbohydrates are basic source of energy. Proteins provide material for growth and development. So when proteins are spent, the growth is spared. Thank you.